So I'm trained as a general surgeon, but I made a lot of my advances through the development of fiber optic endoscopes and laser-based technologies. That was early in my career. Sort of in the middle of my career, we expanded those applications to minimally invasive surgery. And as the Amby Award states, that I really focused on the application of tools and devices for improving healthcare through minimally invasive surgery while demonstrating that they could also lower cost and provide improved access. And this is the key. I believe that technology can both improve healthcare while lowering cost while providing greater access. And so while initially a lot of healthcare technology advances may seem expensive, if we develop them, if we have the fortitude to stay with them and understand that we must appropriately address their failures. Technologies don't move in a straight line. They often have failures and then successes and then failures and successes. That give and take, that ability to learn from your mistakes and make improvements. And there have been many, whether that be in MRI and CT scanning and PET scanning or in angioplasty and stents. All of these devices have had initial successes, then failures, then successes. We learn by mistakes. The key for us today is to make those mistakes in the laboratory and not on the patient. To learn from simulation, to learn from our ability to study these things in silico, in simulation in the laboratory before ever getting near the patient, so we make better devices and can improve them in the laboratory before they ever touch the patient. Then study how they perform in the patient and go back to our simulations and make even better improvements. So in my own career, I've had the ability to do this on a number of technologies. So I was very active in developing miniature fiber optics for endoscopy throughout the body. We started out in looking in the small vessels in the legs and the heart, so-called coronary arteries and peripheral vessels. But we then applied that into the fallopian tubes, the tear ducts, the breast ducts, a number of places where previously man had never looked inside, except at the time of autopsy. And we found that we could see all sorts of pathology that had not previously been recognized, except at the time of autopsy. And that gives us new insight not only into the anatomy, but the pathophysiology of how these diseases arise. It's fascinating to understand that how you view anatomy also changes how you perceive physiology. And it's that cycle of understanding structure and function that I think is important to realize that the ways we perceive things also changes the way we think about their treatment and their cures. So once you can see something, you can treat it. And that's been the whole basis of minimally invasive therapy. If I can see what's beneath my surgical table, if I can see below the surface of the skin and find the tumor, find the disease treatment, and not have to put my hands inside your abdomen or inside your skull to treat your brain tumor or your gallbladder. Now, early on, our group at Cedars-Sinai was very much involved at that time in developing a laparoscopic cholecystectomy, the removal of the gallbladder with so-called laparoscopic instruments, some people like to call it keyhole surgery. And that was enormously successful because it replaced a big open surgery with a small, minimally invasive surgery. We went from patients spending 5 days in the hospital to going home in 24 hours and over a span of just a few years. The same was true in the development, not only in our institution, but many, many others. And here we were not the leaders, there were many others, in the development of balloon angioplasty. Where we tried to advance the technology was in the use of lasers to open block coronary arteries. And while not nearly as successful as balloon angioplasty, it helped develop the technology of interventional cardiovascular disease treatments that has led to this broad array of stents and other devices to open block coronary arteries and block peripheral arteries. And there is a proliferation of these devices, several of which have really improved the health care of many, many hundreds of thousands of patients. In order to make these advances in healthcare, the government has had a critical role. Now, I get to speak in front of public groups on a number of occasions, and they always ask, well, why doesn't industry fund this? And industry does fund the development of the later stages. But basic research, and by basic research I don't mean the material science research, I mean the development of the actual prototype devices, the putting together of the first of a kind prototypes to test in the laboratory. That is done at the academic level, occasionally by industry, but it needs incentives because it is high risk, high reward. And that's where the NIH comes in. This is where it's critical to have government funding. Now, I have been on the halls of Congress repeatedly, and I asked by many congressional staff and congressmen and congresswomen, why doesn't industry fund this? And the answer is industry cannot afford to fund this. If industry were funding basic research, their shareholders would be in an uproar. They would say, why are you wasting money on all this stuff that doesn't have a significant return on investment? Because this type of research is a hit or miss. And the congressman often comes back, congresswoman comes back and says, well, why should the federal government fund it? And the answer is the return on investment long term is enormous. Whether it be the Human Genome Project, whether it be development of efficient batteries, whether it be the development of pacemakers, heart valves, lasers for laser angioplasty, in my case, angioscopes, any number of projects, the investment has been almost always justified. And then they bring up examples where the investment hasn't been justified. And yes, there are plenty of examples where you can find or pick one where that particular investment went bust. Well, you know, that's the nature of research. And anybody who fails to realize that has their head in the sand. You're not going to be successful 90% of the time or even 80% of the time. But you only need one or two successes to hit the ball out of the park, to have spectacular improvements in human health care. And the best example I can give you of this is the pacemaker. You know, this is a tremendous example that has saved millions of lives. And it came about because a gentleman decided that he would try something new, and that story is well known and led to the founding of Medtronic, a multi-billion-dollar industry. Along the way, government has had a major role in developing improved pacemakers. And so has the space project and the technology transfer from NASA, and all the technologies that have led to the miniaturization of the electronics that go into pacemakers, and the development of batteries, and the material science that has helped us make better leads and better batteries and better cases, etc. The host of threats we face and the host of diseases that need solution cost us hundreds of billions. Just look at Alzheimer's disease, or look at threats from viruses. The Ebola scare, if that hasn't awakened some fears in people, I don't know what will. Not that we should be afraid of it, but we have the technologies to deal with these threats. Now, the cost of dementia in the American public will reach billions of dollars a year, every year, well into the foreseeable future. And if we don't learn to cope with this, if we don't learn to understand the causes of the disease, how to slow the progression of the disease, it will cost us tens of billions a year more to the American economy. And the cost and suffering it produces in families is enormous. There are literally hundreds of diseases that could be addressed. And what I mean by that is we can either choose to treat them on the therapy side or hopefully prevent them. Prevention is a far better state than therapy because prevention generally is cheaper. But if we don't invest in the research, we will never get to the prevention stage. And much as we've done with measles, mumps, and rubella and those, we need to have the vaccines available, and we need to educate the public that those vaccines are critical for them to take and not listen to the nonsense about, and I specifically use that term nonsense, that these vaccines are dangerous. There is risk in every decision one takes, whether it's driving across the street or taking a vaccine. But it's a risk-reward benefit. And we must understand that funding the NIH, funding the NSF are critical to supporting continued drivers in the economy. I got off track there a little bit.
With the anti-vaxxers, but I was hoping you'd say that. The dementia case is really compelling.
Yeah, so let me go back to that.
Sure.
So a perfect example of the reason for the need for increased funding is the amount of money that is currently spent on diseases which lead to dementia or mental defect. Parkinson's, dementia, Alzheimer's. These are a group of diseases that cost our economy in excess of $30 billion a year. And yet we spend relatively little treating it or learning how to prevent it. This is, to me, insane. We spend enormous amounts of money caring for all these poor people, and yet we spend little money trying to understand them or treat them. Now, President Obama has tried to start an initiative with $100 million. $100 million to treat a disease of this complexity is a drop in the bucket. It barely scratches the surface of a disease of this complexity. Understand that the human brain has billions and billions and billions of cells that are all highly interconnected. It's like trying to take apart a supercomputer with a toothpick. And it's a small toothpick against a very, very large organism. We need more research that focuses on how the brain is connected, how the brain is organized, and how the brain functions. And only now are we beginning to develop those tools that allow us to understand that structure. It has only been in the last 10 years, with the development of fMRI and optical networks and optogenetics and a whole host of other tools, from PET scanning to fMRI, that we can begin to at least gain some insight on how the brain works. With these tools, and hopefully others that are developed in the future, we can begin to understand how the brain is affected by these diseases and look at the genetics of perhaps what causes these diseases or why we become susceptible to these diseases. These cures are not going to happen overnight. Understanding these diseases will not take place in 2 years or 3 years. But if we keep at it, science proceeds slowly, slowly, and slowly, and then suddenly there may be a breakthrough, a new technology, a chance observation that leads to a critical breakthrough. And it is important to keep the funding levels at a significantly higher level than they are now so that we can accelerate that process. So biomedical engineering is becoming one of the main drivers of our economy in the United States. This is evidenced by the number of students trying to get into the field across the universities in the United States. It's evidenced by the growth of companies doing biomedical engineering and the number of patents filed in this area. These are critical metrics or measures of how successful this field is becoming. But more importantly, it is providing a whole range of new economic drivers for the economy. There are a whole host of new classes of drugs. They're not really drugs, they're biologics, if you will, but they are drugs in the vernacular that didn't exist before. These are materials that are synthesized through the control of cells, be that bacterial or mammalian, that allow us to create molecules we couldn't create before. There is a huge range of biomaterials that we are synthesizing using biologic mechanisms, not enzymes per se, but enzymes within bacteria or enzymes, plant cells or enzymes, animal cells, that allow us to create things we couldn't possibly create. These new drugs, these new proteins, these new biomaterials have enormous potential implications for the treatment and alleviation of pain and suffering, for the treatment of cancer and the treatment of heart disease. And at the same time, we are making much better progress in understanding diseases because our ability to make measurements, to understand how the disease processes are progressing and understand the chemistry of these diseases, what's actually happening at the molecular level, is improving. So biomedical engineering is that intersection where we bring together the physics, the chemistry, the biology, all those different basic sciences to study not just the human organism, but the biologic organism and how they interact. And it may be for medicine and for the biomedical engineering, but it may be also for biological engineering, for a yeast plant or a tree. These are all important in understanding our larger ecosystem. And I want to stress that it's important to fund not just the human research, but the research that goes on at the plant and cell level, because all these things affect how we live in our environment. For years, NIH budgets have been at the discretion of Congress, and this inability to plan makes it very difficult for NIH directors and managers to plan for long-term science. This is a significant drawback, particularly in the conduct of clinical trials. And while they can do some clinical trials, it becomes very hard and very difficult to envision long-term clinical trials when the funding levels vary widely from year to year. More importantly, NIH funding is discretionary, whereas other parts of the budget, such as the Department of Defense, are non-discretionary. This has important implications as legislation makes its way through Congress. Non-discretionary legislation means that it's always going to get funded, and the agencies can count on some minimum amount of money, usually typically what was equal to the year before, maybe a little more, a little less. Whereas discretionary funding, they can't make that budget commitment from year to year to year. Many students, as they enter college, ask me if medical and biological engineering is a good preparation for medical school. And I tell them it's a fantastic preparation for medical school. Because much of medicine is about solving problems. And engineering is also about solving problems. And if you are trained as a medical or biological engineering, your brain works to solve problems. You are trained to solve problems. Now, you don't solve them necessarily in the same way, but that training, that ability to take information in, develop a hypothesis, develop a fundamental theory of why things are working, and then use that to solve the problem becomes very helpful as you move forward in a medical career. And more importantly, once you get into the medical career, it leads you to think of how you can solve the problem better. What can you do to improve the situation? And engineers, if nothing else, are tinkerers. They love to make things better. They love to look at a situation and say, how can we make this better? How can we do this in a better way, perhaps more effectively, perhaps more cost-effective, perhaps with lower complications? But medical and biological engineering is a marvelous background for anyone thinking of a career in medicine.